Hello everyone, my name is Dr. T.S. Kurne, Department of Statistics, Rajarshi Chhatrapati Shahu College, Kolhapur. In last two lectures, we have discussed designs of experiments. First lectures, we have see the basic terminology of designs of experiments, and in second lecture, we have see the principles of designs of experiments. Then now in these lectures, we have discussed which how to done the experiment. So first we have to take the trials. So first terminology, uniformity trials. How to take the trials? Uniformity trials. We have to take uniform trials. So while performing the block, it is an important to know the direction of heterogeneity. Further, to implement principle of local control, direction of heterogeneity plays an important role. Here only important part is the direction of heterogeneity. Direction of heterogeneity which is on row wise or block wise. So we have used the local control to reduces the experimental error. Sometimes on the basis of prior knowledge it is known, but if it is not known, one has to go for the uniformity trials. In any experiment we have used to take the trial. In the field experiment, the fertility of soil does not increase or decreases uniformly in any direction, but it is distributed over the entire field in erratic manner. So uniformity trials gives an idea about fertility variation of the field. Uniformity trials means trials in which the field means experimental material is divided into small units or plots, and same treatment is applied to each of unit and their yields are recorded. From this yield we can draw fertility control map, and which gives a good idea about the nature of soil fertility variation. And this fertility control map is obtained by joining the point of equal fertility through the lines. And the uniformity trials gives us and some idea about the shape and size of the plots to be used. So this is our basic diagram. This whole part is an experimental unit. Make as a homogeneous group, it is a block, and this one block is divided into next it is a plot. So this is the one block, second block, third block, fourth block, and each block we have then partition into 3 plots. So how to make the block or how to make the plot, what is the size or shape, so discuss one by one. But how to decide size of the plot? The size of the plot is depend on the number of factors such as total experiment area available and the number of treatments and number of replications of each treatments of crop, as so on. If the total experiment area remains fixed, then an increases in the size of plot, consequently decreases the size of the block increases. Then there is a decreases in the number of block. Also decreases the number of blocks, then there decreases the number of plots. And Fairfield-Smith of the 10 an important empirical relationship between the plot size and the plot variance after conducting the uniformity trials of the experiment. The relationship is also known as a Fairfield-Smith variance law, and it is given by Vx is equals to V1 divided by x raised to b, taking the log on both sides. So it get log Vx is equals to log V1 minus b log x, where x is the size of the plot and Vx is the variance of the area field per unit area from plot size x unit. And this is the V1 is the variance among the flowers of the size, and B is the regression coefficient indicating the relationship between the adjacent unit. And how to make the shape of the blocks and plot. Shape of the experimental unit or loss mainly depend on the shape of experimental material, particularly in field of experiment. And the clouds are taken to be rectangle shapes. Firstly, the experimental material should be divided into blocks, and the experimental material within the blocks should be homogeneous, and there should be heterogeneous between the blocks, and the number of blocks depends upon the variability pattern of the experiment. Thank you.